Imagine your home if you just, stopped taking out the trash for a month.
Oh, wow. Yeah, that's a rough image.
Right. Just picture it. The bags are piling up in the kitchen. They're spilling over into the hallway. The smell is awful. And there's this complete inability to walk from one room to another.
Exactly.
And eventually, the entire house just completely stops functioning. Now, imagine if your brain cells did the exact same thing.
It is a really visceral image, but honestly, it's a highly accurate one. Because a cell that is drowning in its own waste is, well, it's a cell that simply cannot survive.
Welcome to today's deep dive into the source material. I'm your host today, and we've got a fascinating mission ahead of us.
And I'm the expert guiding us through the science. Thanks for having me.
Of course. So we are looking at a really massive, groundbreaking study today. It focuses on a microscopic garbage disposal system inside your cells.
Yeah, and there is a giant, frankly, intimidating medical term for what happens when this system breaks down neurodevelopmental proteasomopathies.
Right, which is quite a mouthful.
Yeah.
But okay, let's unpack this. What it really boils down to is a look at a specific gene called PSMC5.
Exactly.
And this gene, it acts as a crucial gear in that cellular trash system. So today, we are going to explore how tiny genetic typos in this microscopic shredder can completely alter human brain development.
Not just the brain, too. It reshapes physical features. And it even triggers these massive phantom alarms in the immune system.
It's wild.
It really is. And our journey today is going to take us from the microscopic anatomy of cellular machinery directly into the genetics of 44 unique patients.
Right, and we'll even look into the brains of mutant fruit flies, which I'm excited about.
Oh yeah, the flies are fascinating. And finally, we'll look at the potential therapies that are hiding in our own immune response. It is just a brilliant example of how one broken microscopic gear can halt the entire clockwork of a human being.
So let's just jump right in. Let's start by looking at the anatomy of this cellular shredder. The researchers, they focus heavily on the ubiquitin proteasome system, or the UPS.
Yes, the UPS. It's the central recycling plant of the cell.
Right. And to make sense of this, I like to use an analogy. In our cells, when a protein is damaged or maybe misfolded or just old, the cell tags it with a molecule called ubiquitin.
Perfect analogy.
Ubiquitin is basically like a molecular kick-me or, a trash post-it note attached to a defective protein.
That is exactly the way to visualize it. Ubiquitin flags the trash, but the actual shredding of that trash, that is handled by a massive multi-subunit complex called the 26S proteasome.
The 26S proteasome. Okay, break that down for us.
Sure. You could picture it as having two main components. First, there is the 20S core particle.
The core, okay.
Right, and this is the actual shredding chamber. It's this barrel-like structure that slices proteins down into reusable amino acids.
Got it. So it's the blades of the shredder.
Exactly. But a protein cannot just wander into the shredder on its own. It needs to be recognized, unfolded, and literally pushed in. And that is the job of the 19S regulatory particle.
Which sits on top of the barrel.
Yep, it sits right on top like a cap.
Right, and this cap is made of a lid and a base. And the lid, from what I understand, it recognizes that ubiquitin trash post-it note we talked about.
That's right, the lid checks the tag. But the base is where the real heavy lifting happens. This is where our gene of the hour, PSMC5, comes in.
Okay, so what exactly is PSMC5 doing in the base?
Well, the base contains a ring of 6 molecular engines. They are technically called AAA plus ATPases.
AAA plus ATPases, right.
Yeah, but for our purposes, just think of them as an engine block. There are 6 of them, PSMC 1 through 6. Our focus, PSMC5, which is also sometimes called RPT6, is one of these 6 engines.
Okay, so it's a motor.
Precisely. It utilizes cellular energy, ATP, to physically pry apart and unfold those crumpled, tagged proteins.
Wow.
Think about the sheer mechanical force required here. Proteins are tightly folded, really complex 3D structures. The engine has to literally yank them in.
Like unraveling a tangled ball of yarn.
Exactly like that. Unraveling them so they can actually fit through the very narrow opening of the shredding chamber.
I picture PSMC5 as this, highly muscular bouncer standing at the door of a nightclub. But instead of throwing people out, he is grabbing the trash, uncrumpling it, and forcing it down a chute.
That is a very accurate, very active bouncer.
But okay, here is my pushback on this. If this is just a general cellular trash can, why does a mutation here cause a specifically brain-related developmental disorder?
That's a great question.
Because, doesn't literally every cell in the human body have trash it needs to clear?
Yes, every single cell does rely on this system. But neurons, they're uniquely vulnerable to it breaking down.
Why is that?
Think about how a neuron works. It communicates via synapses, right? Those tiny gaps between cells.
Right.
Well, a single synapse contains hundreds of different proteins. To form a memory or to learn something new, the neuron has to constantly build new protein structures and destroy old ones. It is a highly dynamic construction zone.
Oh, I see. So it's constantly remodeling.
Exactly. It's called synaptic plasticity. If the cellular bouncer, PSMC5, stalls out, the trash piles up instantly, and the synapse completely fails to adapt.
So learning and development basically just grind to a halt.
They do.
It makes you realize that adapting to change isn't just, a mental hurdle. It's a literal physical teardown process in your brain.
Yeah, it really is.
I mean, for you listening, think about a time you tried to learn a new complex skill like playing the piano or speaking a new language. You have to wonder how much of that learning relied on your brain's microscopic ability to just clear out old, inefficient neural connections to make way for the new ones.
Our highest cognitive abilities rely heavily on basic microscopic waste management.
Which brings us to the human toll when that management fails. The researchers identified 44 individuals in this study.
Right, 44 patients.
And they all share this rare neurodevelopmental disorder, and they all carry mutation in this PSMC5 gene. There were 26 distinct variants found among them.
It is really crucial to understand the nature of these mutations, too. The study highlights that the vast majority are what we call de novo missense variants.
Okay, let's break that down. De novo meaning spontaneous, right?
Exactly. It wasn't inherited from the parents. It is a brand new typo that occurred just during early embryonic development.
And missense.
Missense means that just one single amino acid letter in the massive DNA code was swapped out for another. Just one letter.
Just one letter out of billions, and it changes everything.
It really does. And what's fascinating here is where these typos occur on the protein. The researchers structurally mapped the mutations, and they found a hotspot.
Like a specific zone where things go wrong?
Yes. Most of them cluster right in that engine domain we talked about, the AAA plus AT pace domain, the very mechanism that burns energy to unfold the proteins.
Oh, wow. So hitting the engine directly.
Exactly. For example, there is a highly recurrent variant they identified called ARG325TRP.
Right, I saw that in the paper.
Yeah, this typo happens right in the engine, and it causes the most severe multisystemic issues in the patients.
Whereas other variants are less severe.
Right. On the other hand, variants located way at the beginning of the protein outside the engine, they tend to cause milder symptoms. Sometimes maybe just shorter physical stature, but not the severe cognitive impacts.
But when you look at the 44 patients with the severe mutations, I mean, the clinical phenotype is staggering. It's not just a subtle learning curve.
No, not at all.
95% of them have global developmental delay. 81% have intellectual disability. We're talking about a cascade of challenges here. Severe motor delays, autism spectrum profiles, ADHD, and for many of them, active seizures.
It's a profound neurological impact.
But here's where it gets really interesting to me. This is a syndromic condition. It's not isolated to just the brain.
Right.
These kids have skeletal malformations. They have cardiac abnormalities, hearing loss, feeding issues. Why is a brain disorder causing bone and heart problems?
Well, because while the neurons are the most sensitive, that proteasome shredder is literally everywhere. Every tissue needs it, especially during the critical window of embryonic development.
Ah, okay.
And this widespread impact creates physical signatures on the body, particularly the face.
But from what I read, human doctors couldn't actually spot a unified syndrome just by looking at the patients, right?
Exactly. The physical manifestations were too subtle and too varied across the 44 patients. A doctor couldn't just look and say, this is PSMC5 syndrome.
So what did they do?
The researchers used an AI facial analysis tool called Gestalt Matcher.
Yes. This part of the study completely blew my mind. From what I understand, this Gestalt Matcher AI isn't just like looking at a photo and comparing it to a textbook picture.
No, it's much more advanced than that.
It's literally converting a child's face into a massive 512-dimensional mathematical map, right? It plots out every microfeature into a vector space to find these microscopic similarities between patients.
That's exactly how it works. Historically, clinical geneticists relied on their own visual experience, flipping through textbooks, to match a patient's facial features to known syndromes. It's a highly subjective art.
Right, very dependent on the doctor's experience.
But Gestalt Matcher digitizes that process. The AI calculates the mathematical distance between those different patients' facial maps in that 5 and 12 dimensional space.
So the lower the distance, the higher the resemblance.
Precisely. And when they ran the AI on 15 of the patients with PSMC5 variants, it found a hidden facial gestalt. Like
a unified whole that we couldn't see.
Exactly. It's a shared mathematical similarity that human eyes just missed. Subtle things like microcephaly, a slightly smaller head size, or highly specific abnormal jaw structures.
That is incredible.
It is. The AI proved that this specific genetic typo literally sculpts the face in a predictable way.
It is amazing to think about for the listener. I mean, if an AI can see a genetic diagnosis hidden in the shape of a face that trained human doctors completely miss, you have to wonder how this might change the future of diagnosing rare diseases.
It's a game changer.
We might be looking at a future where, like, a simple smartphone photo in a pediatrician's office could instantly flag a rare genetic condition for targeted testing.
Which would save years of diagnostic limbo for these families.
Absolutely. So to push that understanding further, to see exactly how that genetic glitch causes the damage in real time, the researchers had to recreate it in living brains, right?
Yes. And they started with fruit flies, Drosophila.
Fruit flies, okay.
They genetically engineered these flies to knock down or suppress their version of the PSMC5 gene, which in flies is called RPT6.
And then they put these flies through behavioral tests.
Right.
First, they tested classical learning, basically teaching the flies to associate a specific odor with a mild foot shock. And the flies with the suppressed gene learned this perfectly fine.
They did. Their basic initial memory worked.
But then they tested something called reversal learning.
Yes. This is where it gets tricky for them.
I like to use a board game analogy for this. Normal learning is like teaching the flies the rules of a brand new board game. They learn it fine.
Right.
But reversal learning is when you change the rules halfway through the game. Suddenly, what used to be a good move is now a bad move.
Exactly. The flies had to unlearn the first association and learn that a new, different odor meant a shock.
And what happened?
The flies without the fully functioning PSMC5 engine failed this completely. They showed a total inability to adapt. Their behavioral flexibility was just gone.
Wow.
Yeah, this proved that while you can make initial memories without a perfect cellular shredder, you absolutely cannot remodel or overwrite them.
And it wasn't just learning either. They put the flies in a climbing assay to test their motor skills.
Right, climbing up a tube.
Yeah, and early in life, the mutant flies climbed the walls of the tube just fine. But at 22 days old, their motor skills just dropped off a cliff.
Which is really significant. It was a progressive, age-related decline.
So it gets worse over time.
Exactly.
Yeah.
Which gave the researchers a huge clue about how this disease progresses. But to see what's happening at the actual synapse level, they had to go even deeper. They moved to rat hippocampal neurons in a petri dish.
Rat brains this time.
Right. When they suppressed PSMC5 in these rat neurons, the protein trash piled up massively in the cell body. But crucially, they looked closely at the synapses themselves.
What were they looking for?
They stained for two different markers. One marker, V-GLUT, tags excitatory synapses, the ones that stimulate the brain.
Okay, the go signals.
Yes. And the second marker, V-GAD, tags inhibitory synapses, the ones that calm the brain down.
The stop signals. And the results here were wild. The number of excitatory synapses, those go signals, dropped significantly, right?
Yeah, it's a massive drop.
But the inhibitory synapses, the stop signals, stayed exactly the same.
Which is a huge problem. This creates what neuroscientists call an excitation inhibition, or EI, imbalance.
An EI imbalance.
Right. When we talk about an EI imbalance, think of the brain like an intricate orchestra. You need the loud, energetic sections, the excitatory signals, to drive the melody, to process sensory input, to form thoughts.
Yeah, you need the energy.
But you desperately need the quiet, inhibitory signals to keep the tempo, to prevent total chaos.
Right.
If you lose your excitatory synapses, the brain loses its drive, its ability to quickly adapt to new information. This specific type of mismatch is exactly why you see profound sensory processing issues or even autism profiles.
Or the seizures, right, where the electrical activity just cannot be properly regulated.
Exactly. It's a structural imbalance.
They also tested mouse neurons, where they overexpressed the mutated human genes. And they found that some of these mutations actively stunted the physical growth and branching out of the neuron's arms, the neurites.
Yes, neurogenesis was severely impacted. The cells physically couldn't grow properly. It proved it was a loss of function effect.
It really drives home the point that your daily mental flexibility relies heavily on your brain's cellular recycling staying perfectly balanced.
It absolutely does.
I mean, for the listener, have you ever struggled to break an old habit to learn a new one? It might literally be a biological traffic jam in your neural pathways, with the old connections just refusing to clear out.
And that physical traffic jam is at the heart of the cellular domino effect this mutation triggers. We know the bouncer is broken, but the researchers wanted to know what was actually going wrong inside human cells.
Right, because up to now we've talked about flies, rats, and mice.
Exactly. So they looked at human T cells taken directly from the blood of the patients.
And here they found a major surprise. I mean, I would have expected that these typos cause the PSMC5 protein to just degrade, leaving the cell without enough of it.
That would be the logical assumption.
But the amount of the protein in these patient cells was totally normal. The healthy copy of the gene they inherited from their other parent was making enough of it. So if there is plenty of the bouncer protein around, why is the shredder failing?
It is an assembly problem.
Assembly, like putting it together.
Yes. The mutation changes the physical shape of the protein just enough that it cannot properly incorporate into the fully assembled 26S proteasome complex.
Oh, I see.
It's like having all the parts for a car engine, but one gear has a slightly warped tooth. It just won't click into place, so the shredder never fully assembles.
And that kicks off a catastrophic domino effect inside the cell. Because the shredder is broken, the trash piles up into these toxic clumps called agresomes.
Right, agresomes.
So what does this all mean for the cell's survival?
Well, domino number one is that the cell experiences severe proteotoxic stress. And this stress directly damages the cell's power plants, the mitochondria.
Ah, the mitochondria.
Yeah. The researchers saw a massive uptick in a process called mitophagy.
Mitophagy, meaning eating the mitochondria.
Essentially, yes. It's the emergency targeted destruction of mitochondria. Because mitochondria aren't just power plants, they are incredibly sensitive to the environment around them. When toxic protein clumps form, they physically interfere with cellular operations.
Yeah.
The cell senses this toxicity and basically initiates a scorched earth protocol.
Wow.
The cell is literally eating its own power plants because the protein trash is poisoning them.
That is intense. That's domino number one. Domino number two is sheer chaos in the cell's fats.
Yes, the lipidomics data was fascinating.
The researchers ran a profile looking at all the lipids in the cell, the structural lipids that make up the cell's physical borders, the phospholipids. They crashed.
They dropped significantly, yeah.
Meanwhile, cholesterol esters spiked by 50%. The very structural integrity of the cell membrane was just completely destabilized.
It's falling apart at the seams.
Yeah.
But domino number three, the most shocking finding, is what happens to the immune system.
Right.
If we connect this to the bigger picture, we see something called a sterile type I interferon response.
Okay, a sterile type I interferon response. From what I read, normally your cells release interferon when they detect a viral infection.
Yeah. Exactly. It's the body's antiviral panic button.
It causes inflammation, triggers immune defenses, all that. But in these patients, their T cells were pumping out massive amounts of interferon, acting like they were fighting off a severe virus, even though there was zero infection.
It was a complete phantom alarm.
Why on earth is a broken trash can triggering a viral alarm?
It all comes down to a mechanism called the integrated stress response, or ISR.
Yeah, ISR.
Inside the cell, there are specific sensor proteins, like kinases called PKR and GCN2.
PKR and GCN2.
Yeah, and they act as molecular smoke detectors. When they sense that protein trash is piling up, and when they sense the mitochondria dying, they get triggered.
So they trip the alarm.
They hit the panic button, which forces the cell to pump out interferon. The cell literally misinterprets its own internal mechanical failure as an invading pathogen.
That is just incredible. But the researchers didn't just find the alarm, they actually found a way to turn it off, right?
They did. They applied specific experimental drugs like C16 and A92 to the cells to block those molecular smoke detectors, and the alarm stopped.
Wow.
They even used a drug already on the market called baricitinib, which is a JK inhibitor, and it completely silenced the immune panic.
Baricitinib. So this points directly to a potential future therapy.
Absolutely. If we can quiet this phantom immune response, we might be able to alleviate some of this severe multisystemic symptoms in these patients. It really blurs the line between an external threat and an internal mechanical failure.
That's fascinating. And the ultimate proof of all this, it came at the climax of the study using stem cells.
Yes, induced pluripotent stem cells, or iPSCs.
Right. They introduced the severe ARC325T mutation into stem cells that can basically become any cell in the body. And when they forced these mutant cells to form the 3 basic body layers, they formed perfectly fine.
Which is really surprising. It seems these early embryonic cells can somehow compensate for the broken proteasome.
But the fatal flaw only reveals itself later.
Right. When the researchers coaxed those stem cells to differentiate specifically into neural progenitor cells, the direct ancestors of brain cells, they completely failed.
They couldn't do it.
No. They failed to express key neuromarkers like SOX2 and nestin, which are the essential genetic switches required to build a brain cell.
It's like the cells just hit a developmental brick wall exactly when they tried to become a brain.
Exactly. The window where the brain forms is exactly when this rapid protein recycling is needed the most.
It really forces you to reconsider how you view inflammation in your own body, doesn't it? I mean, knowing your cells can mistake their own internal trash pile for a viral infection.
It is a profound shift in perspective, for sure.
So let's bring this all together. We've traced how a single spontaneous amino acid typo, just one wrong letter, breaks a cellular trash compactor.
Yep.
And we've seen how that microscopic failure ripples outward. It alters the growth of synapses, it literally reshapes the bones of the face, and it sets off a blaring phantom immune alarm.
And this raises an important question. Well, a really provocative thought, actually.
Yeah.
And it is perhaps the most profound takeaway from the entire paper.
Okay, let's hear it.
Let's look at the cellular signatures we just discussed in these young children. Broken shredders, protein trash aggregates, mitochondrial death, sterile immune inflammation, disrupted cholesterol.
All the dominoes. Wait, are you saying...
And those are the exact same cellular hallmarks we see in aging brains and neurodegenerative disease.
Oh, wow.
We see this in Parkinson's, we see this in Alzheimer's, we see this in Lewy body dementia.
So a rare neurodevelopmental disorder in young children is literally mirroring the biology of a neurodegenerative disease in the elderly.
Precisely. The core mechanical failure is identical. It is just happening at the very beginning of life instead of at the end.
Got it. That's incredible. Which leaves us with a really provocative thought for you to end on. Could understanding and treating this rare, tragic childhood developmental disorder actually hold the hidden blueprint for curing the neurodegenerative diseases that plague us in old age?
It's a massive question.
If we learn to fix the bouncer and quiet the alarm for these kids, could we do it for an aging brain?
We just might be able to.
Well, thank you for joining us on this deep dive into the source material. The microscopic universe inside you is vast, intricate, and deeply interconnected. Keep questioning it.